we are going to study about the typical stamen. Typical stamen consists of three parts, that is the filament, then anther lobe, and both the lobes are joined with the help of connective tissue. So these are the three parts. Stalk of the anther is called filament, and on the filament there is resting of anther lobes. They are bilobe structure. It is bifix, and both are joined with the help of connective. And after maturity, there is dispersal of pollen grain. The next diagram is TS of anther. In this transverse section of anther, when we take the transverse section, it consists of 4 pollen mother cells, and that is called the 4 pollen sacs. And 4 pollen sacs are covered by the 4 layers, that is the epidermis, then endothecium, middle layer, and tapetum. First one is epidermis, that is the epidermal part. It is protective in function. Then endothecium is the second layer. It helps in dehiscence of anther. And then third is middle layer, supplies the nourishment to this sporogenous tissue. And last is tapetum, provide the nourishment to the pollen grain. It also helps in formation of enzyme of pollen grain. It also secretes special type of protein which helps to decide compatibility of the pollen grain. Next diagram is pollen grain. Pollen grain, it is haplod in nature, it is uninucleate, it is unicellular, it is spiny, and it consists of exine, intine, and nucleus. So in this pollen grain, development of ovule is called microsporogenesis. Pollen grain or microspore is haplod in nature. It is first cell or male gametophyte. Wall of the pollen grain is called sporoderm. It consists of outer exine and inner is called intine. On the exines are present germ pores. Intine is made up of pectin and cellulose. Exine, etapcutinase. It contains special fatty chemicals called sporulinin. It is resistant to bacteria and various chemicals. Due to this, pollen can preserve for a long time in the soil. So, fossils of pollen grains are very common. So, in these pollen grains are monocolpate in monocots with one germ pore. In dicot, they are tricolpate. In insect pollinated flowers, exine is covered with yellowish viscous sticky substance called pollen kit. Exine is absent in aquatic plants. So in this anthesis, dehiscence of anther and release of pollen grain is called gynoecium. It is female sex organ. It is innermost whorl of the flower, innermost essential whorl of the flower, female sex organ of the flower. The condition of gynoecium is stigma, style, and ovary. So in this placentation, arrangement of ovule or placentae inside the ovary is called placentation. There are following types of placentation. First one is marginal placentation. Ovules are attached to one margin of ovary. Example, groundnut. Second is axial placentation. Ovule is attached to the central axis of the ovary. Example, China rose. Basal placentation. Ovules are attached to the base of the ovary. Example, sunflower. And last one is parietal placentation. Ovules are attached to the periphery of the ovary wall. Example, cucumber. Then study about the ovules. Ovules. Orthotropous ovules, there are many types. Orthotropous ovule, this is straight ovule. In this type, micropyle and chalazar are in one straight line opposite to each other. Example, polygonium. Second is anatropous ovule. This is a inverted ovule. It is the most common type of ovule. In this ovule, micropyle and funicle are close to each other. Example, jawar, sunflower, China rose. Cameplotropous ovule, curved ovule. Example, chenopodium. Amphitropous ovule, this is a horizontal ovule. Example, lemma. Cercinotropous ovule, turn at more than 360 angle. Example, opuntia.